Hi, and welcome to the Neurological Foundation's Discovery Sessions. The Neurological Foundation has been funding research into neurological conditions and diseases for nearly 50 years. We have funded some amazing scientists and clinicians throughout their career, including today's speaker, who have gone on to conduct groundbreaking work and discover fascinating things about the human brain. We hope that these digital discovery sessions will help all our followers, supporters, and fellow science enthusiasts learn new things about one of the most complex organs humans have, their brain. I'd like to introduce today's presenter, Dr. Amy McCaughey-Chapman, a postdoctoral fellow of the Catwalk Spinal Cord Injury Test. Amy received a W&B Miller postgraduate scholarship in 2014 and a small project grant in 2018 from the Neurological Foundation. Amy has recently finished her work in developing a unique way to construct 3D human cell models in a dish, which has the potential to advance our understanding of cell transplantation and neurological conditions. We're grateful to Amy for taking time today to give us a glimpse at her innovative work turning human skin cells into human brain cells, and telling us how this process has helped advance our understanding of neurological conditions such as Huntington's disease.
All right, good evening, everyone. So I'm Amy, and as said in the introduction, I am a postdoctoral research fellow in Professor Bronwen Connor's laboratory. I have been in Bronwen's lab for 7.5 years, and the main technology that we use, which I will talk to you about tonight, is cell reprogramming. So I'm just going to share my screen with you all so that you can now see my slides. So I'm going to talk to you about growing brain cells and neurological disease in a dish. In preparing for this talk, I was asked what one of the most fascinating facts that I have learned through my research is. And doing cell reprogramming, which I will talk to you a bit more about shortly, I spend a lot of hours looking after cells, feeding cells, staring at cells. So one of the most fascinating things for me is that neurons are absolutely gorgeous when you look at them down the microscope, but also oligodendrocytes, which I will talk to you a little bit more about towards the end of the talk, are my new obsession, and I think they are absolutely cool. But overall, something that is fascinating and sometimes frustrating for some of us in the lab is that these reprogrammed cells that we work with really have a mind of their own sometimes. But we do find that with great care, attention, and diligence, it is possible to study neurological diseases in a dish. So I thought I would give you a glimpse into what I actually do every day before I walk you through a little bit more of the science. And so you've got a series of photographs here. First one, you can see me sitting at this hood. And this hood just basically allows the cells to remain sterile. So it's protecting the cells from me, and it's also protecting me from the cells. And I spend hours sitting at this hood, and this is how we look after our cells. So you might have seen online or on the news, anytime there's a flash news about a scientist, there's usually this pink bottle of solution is shown, and you've got this, usually a plate with this pink solution in it. Well, the pink solution is just basically media that is made up of amino acids, vitamins, and salts, and we can add all sorts of factors to it, and that is what we give to our cells. And so when we feed our cells, you can't actually see them. They're sitting in the plate here, and all we do is we take the pink solution off and we give them some fresh pink solution. So actually quite straightforward. We spend a lot of time looking down the microscope, and we do so to check our cells to see what they look like, but also to assess whether they have turned into what we try to do. So cell reprogramming, what does this actually mean? what we do is we convert adult human skin cells into brain cells. I've got this little schematic which I have put together. So we purchase human skin cells that are called dermal fibroblasts, and we reprogram them, which just means converting them into human neural precursor cells. So these are just basically immature brain cells. And we do so by delivering two factors that are important during human brain development. The way we deliver the factors is through using chemically modified mRNA, which is non-toxic to the cells and allows for these two factors, SOX2 and PAX6, to enter into the cells efficiently. So we deliver these genes and we combine it with a defined reprogramming medium, which is just the pink media, which I showed you in the photos before. And depending on the protocols in the lab, within 2 to 3 weeks, we have these human neural precursor cells. And then we switch the pink media again to a different media that has different factors that promote neuron generation. And we can then push these neural precursor cells into mature neurons and astrocytes. So we generate a mixed culture in our cultures. So we've got neurons and astrocytes in the same dish. And this whole process is quite fast, although we sometimes feel it's quite a slow process. It really only takes a maximum of about 6 weeks. And the great thing is that in neurological diseases, there are often different types of neurons that are affected. And these different types of neurons come from different areas in the brain. So for example, in Parkinson's disease, dopamine neurons that are found in an area called the ventral midbrain are affected, and we can tweak what is in the medium during this phase so that we generate neural precursor cells that are actually of a slightly more dopamine lineage, so that then they give rise to dopamine neurons, and we can use them for Parkinson's disease. And we can do the same thing for Huntington's disease, as well as generating a more cortical subtype of neurons, which is being used in the lab to model Fragile X syndrome, a common form of autism. So why is cell reprogramming important for neurological conditions? And as I touched on, neurological conditions, which include Huntington's disease, Parkinson's disease, and Alzheimer's, to name a few, they are very common. And sadly, there are basically only symptomatic treatments available. But why cell reprogramming works or has potential is because each of these diseases is linked up to a specific type of neuron or population of neurons. So I have been working since 2018 on a Huntington's disease project. And so I will focus on Huntington's disease for this next part of the talk. So for those of you who don't know, Huntington's disease affects 1 in 15,000 individuals. It is an autosomal dominant genetic disorder. The disease onset is usually between 30 to 50 years of age, and it is sadly fatal. On a cellular level, Huntington's disease is caused by a selective and predominant loss of a specific type of neurons in an area of the brain called the striatum, which you can see here sorry, it went too fast, which you can see here in this photograph of a normal brain. So the striatum is here, and there are neurons in this area that are called GABAergic medium spiny neurons. And in this Huntington's brain, you can see that there is a massive striatal atrophy, and so there is loss of the striatum, and that's because there is loss of these GABAergic medium spiny neurons. So what we've been working on in the lab is to use our cell reprogramming technology to generate GABAergic medium spiny neurons that, as I just said, are important in Huntington's disease. So here's a picture of what skin cells look like. You may find that fascinating. They are just basically elongated, fairly irregular shaped cells, and they often grow at a rate of knots. So here are the skin cells that we've purchased, and we reprogram them, as I said, towards a neural precursor fate, and this is what our neural precursor cells look like. So we've got some single cells, but they also assemble into these balls of cells called spheres, which we also refer to as colonies. And then once we've got these neural precursor cells, then we differentiate them, so mature them towards mature neuron phenotype. And here are what the neurons look like. And then what we do is to check that they are actually neurons and that they are the right type of neurons. We can stain them for two markers. So in this case, in red here is just a marker for neurons, and in green is a marker for GABAergic medium spiny neurons. So when you've got an overlap in the slightly yellowy orangey color here, it just shows you that not only are these neurons, but they're also GABAergic medium spiny neurons. So the relevant cell type lost in Huntington's disease. What's even better is that we can purchase skin cells not only from healthy individuals, but we can purchase skin cells from individuals who have Huntington's disease. So we can essentially generate neural precursor cells and GABAergic medium spiny neurons from Huntington's donors. And so we can essentially have a cell model of Huntington's in a dish, and we do have a cell model of Huntington's in a dish. So there's lots of different applications for this model, but what I'm going to show you next is some of the applications in terms of drug discovery. So because we have these cells sitting in a dish, we can easily manipulate them. We can easily put some compounds on them to see what happens inside the cell at the cellular level. So it's a very good way of testing compounds on cells, because you can just watch. If it kills the cells, you will know that the next day. You will actually know it within a few hours. You can just put the cells under the microscope and have a look. So to take you into a slightly more complex scientific aspect to Huntington's disease, which I will try to keep very simple. This is a schematic of a medium spiny neuron. So a medium spiny neuron, the neuron that is lost in Huntington's disease. This is the cytoplasm, and this is the nucleus of the cell, so the center of the cell. Now, there is a factor called brain-derived neurotrophic factor, or short BDNF, which, as its name indicates, is found in the brain and is very important in the brain for the generation and the support of neurons. Now, in Huntington's disease, brain-derived neurotrophic factor is reduced. And at the cellular level, what you see is in a cytoplasm here, in a normal, healthy individual, you have this Huntington protein. And it is bound to this other factor called REST. It remains in the cytoplasm in healthy individuals. And in the nucleus is where you get BDNF transcription, so that you get protein expression of that factor. And then the BDNF is packaged up into these little vesicles, which are transported down branches of the neurons. And then the neurons pass them across to each other, and that is how it circulates in the brain. Now, in Huntington's disease, the huntingtin is mutant, and that is what is called polyQ huntingtin, because it has an expansion. So when the huntingtin is mutated, the rest is no longer bound to it, so it dislodges, and that means it's free to do what it wants. So it moves into the nucleus. And by doing that, it blocks the expression of BDNF. And that is why there is low BDNF in Huntington's disease. So what we did with our cells is, here you have cells from a healthy donor, and this rest factor, which in healthy cells is found in the cytoplasm, is labeled in green here, and you can see that there isn't particularly a bright spot in the nucleus of rest. It just looks like it's got a green, it's everywhere in the cell, is the way it sort of looks. So it's in the cytoplasm. Whereas if you look at this panel here, which is neurons that have been generated from a Huntington's donor, you can see these bright green spots here, which show that there is rest in the nucleus of the cell, which is exactly what we said happens in Huntington's disease. So our cells are exactly modeling Huntington's disease and doing exactly what they should. So then we thought we would try testing some compounds to see if we can see what happens to rest in response to those compounds. So X5050 is a compound that blocks rest, inhibits rest. And if you compare the green here to the green, the way the green looks in these cells, you can see that the green in these cells looks pretty similar to these ones. And so it has blocked rest. Rest is in the cytoplasm again. Lithium chloride is a compound that has been used for all sorts of things and has been tested in Huntington's disease. And so we put lithium chloride on our cells, the Huntington's ones. And we also saw in some cells that rest was sent back or blocked from moving to the nucleus. It was in the cytoplasm. And so what we are now working on, and I was hoping to have this data ready, but then we are back in lockdown and not able to access the lab, is we're seeing whether the treatment of lithium chloride, the fact that it seems to have an appropriate effect on rest, we're now measuring the levels of BDNF in those cells to see whether it's had a complete effect on the BDNF and whether those compounds could be potentially therapeutic. And there are actually other compounds that I've been working on as well, and that I am rounding off that data, hopefully once we get back into the lab. So a little bit of science here, but hopefully it gives you a little bit of an idea of how we can use our cells, and how just by having access to cells from a Huntington's donor in a dish, it is quite easy for us to basically chuck on a compound and see what happens. So next I'm going to change gear a little bit and talk about 3D modeling. So keep talking about these cells in a dish, right? The cells are essentially three-dimensional, but by having them in a dish with a flat surface, it's causing the cells to basically turn into a two-dimensional flat system. Now, if you think of the organ of the brain, or just yourself, we are definitely not flat, we are definitely not two-dimensional. And there is evidence that cells that have been stretched out across a dish and therefore are flatter could actually be behaving artificially. So what we've been interested in for a little while, and I've been working on, is trying to see how we can create a three-dimensional scaffold, essentially, that we can put ourselves into to still have them floating around the dish so that we can still chuck on our compounds and manipulate them really easily, but that they have a three-dimensional mold that they can grow in that is more closely resembling the brain than a 2D flat surface. So there is a whole field of biomaterials that has done lots of work on what are called hydrogels, which is basically just combining two compounds, often natural compounds, can be synthetic. And this particular hydrogel, which I've done a fair amount of work on and that we have a master's student working on this year, is called GELMA. GELMA, known as gelatin methacrylol, looks like this. So this is, it actually feels a bit like polystyrene, but this is the GELMA compound. And all I have to do is add these two powders to it, and these are photoinitiators. Photo meaning light, which means this polystyrene GELMA, I can get it into solution. So I just add some liquid to it and it turns into a solution. And I can put these powders into solution, as you can see here. And then I can mix them together. And I now have this orange gel, which is not yet gelled. It's just a solution. And I can mix it with my cells. And once I've mixed it with the cells, I can then drop them on into these molds, which just creates a little droplet. And because these two powders are photoinitiators and therefore solidify when you shine light on them, once you've got your cells inside and you've put it in these molds in the plate, you just shine an LED light over the top for 10 minutes. And then you now have this yellow blob, which is your gel with cells inside. And you now have cells in a three-dimensional droplet sitting in the dish. And so what we have been able to already show is that we seem to get better yields of neurons. So we get more neurons if they're in 3D. And what Lynn, our student, is working on this year is seeing whether they survive better. And she's looking at characterizing the type of neurons that generate and seeing what happens as a result of them being in this three-dimensional droplet. So it's a very, very exciting project that's coming along as well. And the last thing in the last few minutes that I will briefly go through is the new work that I have just started. So the Catwalk Spinal Cord Injury Trust is funding me for 2 years to conduct a project, which I'm very grateful for. And I am basically in this project moving away from neurons, which we've been talking about up until now, to focus on oligodendrocytes, which, as I said at the start, are my new obsession. So we call them oligos for short. And if you don't know what an oligo is, here's a schematic I've put together. So here is an oligodendrocyte with these branches. And here is a neuron. Now, these oligodendrocytes produce this insulating sheath that is called myelin. And so just like a wire with a cable, you have the branch of a neuron called an axon that is protected by this insulating layer made of myelin that is produced by these oligodendrocytes. The fact that these axons are myelinated means that the neurons are protected, and they also can communicate fast and efficiently. So if something happens to the oligos or the myelin that's produced by the oligodendrocytes, then you end up with demyelinated axons or branches of the neurons, which means they are prone to damage and degeneration. And they also can't communicate as well. And why am I talking about this? It's because this is highly relevant in spinal cord injury, but also other demyelinating diseases such as multiple sclerosis. So my work is going to be focusing initially on spinal cord injury. Spinal cord injuries affect 2.5 million individuals worldwide, and they are absolutely devastating. Rehabilitation and decompression surgery with symptomatic relief is the treatment at the moment, and oligodendrocytes are susceptible to spinal cord injuries, and they contribute to the pathology. So they're very much involved. The field is basically thinking that to find a complete treatment for spinal cord injury, it will have to be some form of combination therapy approach. So rehabilitation will always be involved. But so what I am setting out to do is to use our cell reprogramming and not generate neural precursor cells like I've talked to you about, not generate neurons like I've talked to you about, but generate oligodendrocyte precursors and mature oligodendrocytes. And I have done this, so I've got some pictures here to show you. So here are, again, the skin cells. And this is what the oligodendrocyte precursors look like. So hopefully you'll see they look a little bit different to the spheres, colonies that we get in our neural precursors. And they've sort of got all these sort of star shapes. And then when I differentiate these to generate mature oligodendrocytes, you get these jobbies, which I think are nothing like neurons, and absolutely, when I first saw it, I thought it was very cool. So this is what an oligodendrocyte looks like. A star or a spider, whichever way you look at it, but they're quite different looking. And so that is what I'm going to be doing for the next couple of years. I'm going to be generating these oligodendrocytes to see how they might fit into some form of combination therapy for spinal cord injury. And hopefully after that also apply them and see what we can do with them in terms of multiple sclerosis. So I will wrap up this part of the talk before we open up for questions, but I would like to acknowledge my supervisor and mentor, Professor Bronwen Connor, who has been an absolutely fantastic mentor for the past 7.5 years. I have to definitely thank the Neurological Foundation and those who donate to the foundation. All this work, as said in the introduction, has been made possible through those donations, either directly funding my scholarships or funding the grants so that we can buy everything that we need to buy to conduct the research. So I am very grateful to those donors and to the money that's been donated. And thank you all for listening, even though I can't actually see you. So we'll now move on to some questions. So the first question that has come through, I will actually stop sharing so that you can hopefully see me. So the first question that's come through is, what type of implications could this cell remodeling and reprogramming have in the progression of neurological research? So neurological research in general, so there's various applications to cell reprogramming. As I covered today, there is a whole side of things in terms of looking for drug treatments. So you can test compounds on the cells. You can see what's going to kill the cells, see what happens inside the cells in response to those drugs. And it's a first platform for drug discovery. You also can simply use the cells as a model. So for some diseases, we don't totally know what's going on at the cellular level. We know what cells are involved, but we don't know what's going on inside. And so that can, by modeling the neurological condition using the cells, you can start to find potentially new drug targets and new therapeutic targets. And then there is, of course, a big field in focused on cell replacement therapy, which means using the cells, transplanting them initially into often rodent models of the neurological diseases to see whether they can replace the cells that have been lost. And then if that works, then moving into clinical trials. And there is a whole field of cell replacement therapy that has been going on since the 1990s and is still going on. So that's the key implication for cell reprogramming. And because in our technology we use adult human skin cells, the concept is you could take skin cells from someone who has, say, Parkinson's disease. You could generate the appropriate neural precursor cells from their skin and then put it back into their brain, and you would therefore not have no immune rejection because it's their own cells. Question number 2. Could this type of 3D modeling for cells be used for other medical purposes, such as growing organs like a liver for transplant? Absolutely. 3D modeling in general is that it's used across the board. It's not only for neurological research or simply the brain. There are some very clever bioengineers who are playing around with, I say play, they may not say play, playing around with compounds, creating hydrogels. There's also a whole field looking at actually generating what are called organoids, which means you allow the cells to assemble into a bigger cell mass which resembles the organ. So for liver transplant, for kidneys, there is definitely a lot of research going on in terms of 3D modeling, be it with the hydrogels, be it through the organoid field. Absolutely, yes. See if there's any other questions coming through. Just waiting for them to actually come through to me. So the next question is, presumably the oligodendrocyte development will lead to animal studies. Recognizing your work is in early development, when do you think there might be animal studies commencing? Well, I like to be optimistic, and I have a very good feeling about these oligos, not only because I think they're cool and I'm obsessed with them, but they seem to be progressing well. So animal studies could possibly be commencing towards the end of next year, if not the following year, it does also depend whether we spend more time in lockdown or whether we can have access to the lab, of course. The next question is, you said you take Huntington's patients' skin cells, which then reprogram into faulty GABAergic medium spiny neurons. Do we know if this basic error apparently in all these patient cells is expressed elsewhere outside the brain? So Huntington, which is what I was talking about in terms of Huntington's disease that's mutated, is ubiquitously expressed. It's expressed everywhere. So the faultiness, if you want, in terms of the Huntington being mutated, it's definitely been looked at in the brain because that's the organ that's studied for Huntington's disease. I don't actually know, but I wouldn't be surprised if mutant Huntington was found elsewhere. The next question is, do you feel this research can also be used to help people with periventricular leukomalacia? I don't know, is the answer to that. Possibly. Next question is, where do you hope the research will lead for those with Huntington's? There's a lot of different things going on in our lab for Huntington's disease. There is the drug testing and discovery component. There is the cell replacement therapy component, which I didn't talk about today, but I touched on the cell replacement therapy field in general. But that is quite promising and prominent for Huntington's disease. And that possibly also linked up with gene therapy work, which Bronwen Connor has done a lot of that previously. We're not focused on it at the moment, but there are other groups who are, and again, it could be a combination approach. So the same way I touched on that for spinal cord injury, I think to actually tackle some of these neurological diseases, it's not just going to be one treatment. It will probably have to involve multiple approaches, involving cells, involving drugs, because they are very complex disorders. Great. So I think that's all the questions. I want to thank you all for listening in. I'm assuming there were some of you listening in because there were some questions coming through, so that was great. I hope I didn't lose you in the science. I hope that gave you a little bit of insight into what we do. And thank you very much for listening in. Goodbye.